This presentation, made by the 2014 Colorado College Biochemistry class, is designed to give a brief background on HIV and AIDS. Our class spent a semester studying the chemistry of the HIV retrovirus under the instruction of our professor, Nina Grover. In the presentation, we will cover the history and progression of the disease since it emerged in 1981, the life cycle of the virus on a cellular level, and finally, we will cover some of the available treatments, prevention strategies, and resources that are available within the Colorado Springs community. We hope that the presentation will give a comprehensive view of the virus in language that can be understood by anyone, regardless of scientific background. The aim is to provide enough information so that anyone who views this video will be able to use it to inform others and spread awareness of this devastating condition.
Today, more than 34 million are living with HIV/AIDS worldwide. Since 1981, when the first case of HIV/AIDS was reported, AIDS has taken the lives of more than 30 million. Unfortunately, 97% of those who are living with HIV are residing in low-income countries, particularly sub-Saharan Africa. In the United States, more than 1.1 million are living with HIV, and the scary fact is that every 1 in 6 who are living with HIV do not know that they're infected. In addition, about 1 in 4 of new HIV infections in the United States is among the youth of ages 13 to 24. Most of them do not know that they're infected, therefore they're not getting the right treatment, and they can spread the disease without even knowing.
From very early on in the fight against AIDS, both U.S. and global initiatives have attempted to educate the population on HIV and its effects as a deadly virus. In Washington, AIDS Action has worked since 1984 to educate the populace and government, as well as to represent those affected with this virus and corresponding autoimmune disorder. Globally, the World Health Organization has had an initiative since 1987 that supports AIDS education and research throughout the world. Nevertheless, many misconceptions and misunderstandings still exist within our society as well as around the globe. For example, since 1985, homosexual and bisexual men have been banned from donating blood because of the stigma associated with HIV. The first misconception is that HIV and AIDS are the same thing. This is not true, however, as HIV is the virus that infects human helper T cells, while AIDS is the acquired disorder related to the inability of the body to recognize and fight invaders due to the inability of helper T cells to undergo their normal cellular mechanics because they are too busy producing more virus. HIV-infected patients do not automatically have AIDS, but can develop it if the virus is allowed to grow unchecked. The second misconception is that people receiving treatment for HIV can't spread the virus. This is also untrue because with current medications, which we will discuss in detail throughout this talk, individuals infected with HIV can be treated to the extent that the viral load in their bloodstream drops to undetectable levels. Although this allows for a healthy, normal life, there is still a small amount of the virus present in the body forever. People who have HIV can still protect themselves from transmitting the disease to others through safer sex practices, however, so once again, the disease is not a death sentence. This leads to the third misconception, that HIV infection is a death sentence. As I just alluded to, since the 1980s there have been drugs on the market that can successfully repress the viral replication system through entry inhibitors, fusion inhibitors, reverse transcriptase inhibitors, integration inhibitors, and elongation inhibitors, which will be discussed in depth later on in this presentation. I won't get into these much here because of this fact, but it is important to note that these drugs do have side effects, and the HIV virus itself can be resistant to many drugs if it has the correct mutations. This leads to the fourth misconception, that which states that since HIV/AIDS can be cured, it's no big deal to get infected. This is not true, however, because despite all current efforts, infected patients will always have lingering virus in their helper T cells. The cells can still function correctly under medication-induced conditions, but to maintain these conditions, a person must take a regimented dose of drugs for the rest of their life. Also, the virus will continue evolving over time as well, and may gain new resistances to existing medications. Drugs also feature negative side effects that should be considered when debating taking them.
The fifth misconception is that if you and your partner are both HIV positive, there's no reason to practice safer sex. In actuality, if you and your HIV positive partner do not practice safer sex, one of you can transmit a mutated form of HIV to the other person, so you can transmit a drug-resistant form of HIV to each other. Another myth is that you can't get HIV from oral sex. Although the chance of getting HIV from oral sex is less than other forms of sex, it is not zero. Risk of transmission is still possible due to cuts or lesions in your mouth, such as cold sores or canker sores, so a latex barrier like a condom or a Dapper Dam should still be used during oral sex. The seventh misconception is that you can get HIV from any contact with an HIV positive person. This is not true. Studies have shown that HIV cannot be spread through touch, breath, sweat, saliva, or tears, and it cannot be spread by touching something after an HIV positive person has touched it, by hugging, kissing, or shaking hands with that person, or by sharing eating utensils or food with an HIV positive person. Another misconception is that if you're straight and you don't use IV drugs, you can't become HIV positive. This is not true. About 16% of men and 78% of women become HIV positive from heterosexual contact and not through same-sex contact or needle sharing. A final misconception is that you will be able to tell if your partner has HIV. This is also not true, as HIV can remain dormant in a body and completely symptomless for many years, but is still transmissible.
In order to understand these facts, it is necessary to place them in their historical context. The 1980s are often regarded as the beginning of the modern AIDS epidemic, as the disease became increasingly prevalent throughout this period. After I discuss some of the significant dates within the 1980s, we will move on to more recent events occurring in the 1990s and 2000s. On June 5th, the Center for Disease Control and Prevention, or CDC, reported cases of a rare lung infection in 5 young, previously healthy gay men in Los Angeles. All these men had very unusual infections as well, which indicated complications within their immune systems. These and other rare cases quickly spread, and by the end of the year, over 270 cases of severe immune deficiency in gay men were reported. On September 24, 1982, the CDC used the term Acquired Immune Deficiency Syndrome, or AIDS, for the first time. In February 1983, the National Institute of Health suggested that a retrovirus is the probable cause of AIDS. The discovery that a retrovirus causes AIDS sparked controversy among 2 groups that each believed they had made the discovery. The first group was led by Dr. Luc Montagnier of the Pasteur Institute in Paris. Dr. Montagnier and his colleagues isolated a retrovirus that killed T cells from the lymph systems of an AIDS patient. This retrovirus was named lymphadenopathy-associated virus, or LAV. At the same time, a team led by Dr. Robert Gallo reported the discovery of the virus, but they renamed it Human T Lymphotropic Virus Type 3. Now, whether Gallo or Montagnier deserve more credit for the discovery of the virus that causes AIDS has been a matter of considerable debate. Dr. Montagnier and one of his colleagues were awarded the Nobel Prize for the discovery. However, Dr. Gallo is also considered a co-discoverer. In May 1986, the International Committee on Taxonomy of Viruses ruled that both virus names should be dropped and a new name, human immunodeficiency virus, be used. On March 19th, the FDA approved the first antiretroviral drug, zidofadine, or AZT. Congress then approved $30 million in emergency funding to states for AZT.
The 1990s were regarded as a decade in which many programs and initiatives were established. In the 1980s, Ryan White, a 15-year-old boy, experienced discrimination because of his status as HIV positive. Then in 1990, the government enacted the Comprehensive AIDS Resources Emergency Act as a means to provide care and treatment for individuals with HIV. Then there was a modification to the Americans with Disabilities Act that would cover individuals with HIV. Then in 1991, the red ribbon was established as the international symbol for AIDS awareness. In 1995, the CDC enacted the Syringe Exchange Program as a means to prevent infectious disease. In 1996, the CDC approved the viral load test. In addition, there was a huge initiative to speed up the discovery of a vaccine for AIDS. Then in 1998, AIDS was becoming more prevalent in minority communities. Thus, President Clinton established the Minority AIDS Initiative. Then in 1999, the CDC revised the definition of HIV infection.
So now that we've heard the history as it happened over the course of the first 20 years of the HIV/AIDS pandemic, I'd now like to speak a little bit about how this virus has affected the world within the last decade or so, as early as 2000 and moving into the 2010s. In the year 2000, beginning with his State of the Union address at the turn of the century, Bill Clinton announced that HIV/AIDS was now a threat to national security. This effort to combat the spread of the disease in developing countries overseas was doubled to $254 million, with high priority being paid to sub-Saharan Africa. Then, in 2002, the FDA, or the Food and Drug Administration, approved the first rapid HIV test, with results being accurate 99.6% of the time and taking only approximately 20 minutes. This was certainly a major breakthrough in terms of getting more people tested for the disease. This test was also to be transported much more easily in the field, as it did not require heavy amounts of machinery, specialized equipment, or extensive training to operate the test, making it extremely useful for widespread testing. In 2003, PEPFAR was established, and this was the President's Emergency Plan for AIDS Relief. George Bush created this program, and it was effectively a $15 billion plan to combat AIDS over a 5-year period. In 2006, this marked the 25th year that AIDS has been around in the United States. And then in 2009, Barack Obama launches the GHI, or the Global Health Initiative. This program initiated a $63 billion effort to develop awareness of global health in low and middle income countries. PEPFAR, reauthorized 1 year earlier for an additional 5 years, is at the core of the GHI. In 2010, the HIV travel entry ban was lifted by President Barack Obama. For 22 years, a ban had been placed on travel into the U.S. by anybody infected by HIV. This ban had for 22 years furthered the stigma around HIV and prevented any non-U.S. citizens from obtaining tourist visas or permanent resident status based on the fact that they had HIV positive. Obama claimed that the ban was a decision made on fear and not on fact, and that if we want to be a world leader in the fight against HIV, we better start acting like it. The lifting of this ban allowed entry of thousands of students, tourists, and refugees, as well as, importantly, adopted children who are HIV positive. Public debate then began in 2011 on whether or not people already infected with HIV should be allowed to receive organ transplants from other HIV-positive people. Also in 2011, Elizabeth Taylor, one of the first celebrities to publicize that she had contracted HIV, passed away. She had formed the AMFAR, or the American Foundation for AIDS Research, in order to help research and develop drugs for the treatment of HIV and AIDS. In 2012, the FDA then approved OraQuick. This was a store-bought, over-the-counter test that uses oral fluid to detect HIV. A swab is essentially rubbed in the inner linings of the cheek, and the test is complete within approximately 20 minutes.
So how does HIV work? We will be going through the virus replication cycle in detail in order to help you understand how the virus infects someone.
HIV can be transmitted through sexual contact or infected blood. Researchers believe that the virus attaches to immune cells called dendritic cells, and they are found in the mucosal membranes. The dendritic cells aid in transporting the virus to the lymph nodes, which interact directly with T cells, allowing HIV to infect T cells. Once a foreign invader is recognized within the immune system, T cells, produced in the thymus, are employed to signal for further responses to occur. HIV targets T cells. This is how HIV knocks down the immune system, because T cells aren't able to do their job because they are destroyed. HIV also uses T cells to produce more viruses.
Now that the virus has infected the human, the host cell needs to bind with the viral particle, as you can see on the left. The viral particles which are floating around have receptors on them which are going to come into contact with the white receptor that you can see on the left on the host cell. As you can see on the right, the GP120 protein binds with the CD4 protein that's on the host cell. These then interact, causing the co-receptor to bind to the host cell membrane. Once the co-receptor binds, this is going to facilitate the fusion of the viral particle and the host cell particle. Now that the membranes have fused and the viral RNA is inside the host cell, it needs to reverse transcribe. Normally, DNA turns to RNA, which turns to protein, but in this case, we're starting with viral RNA. The reverse transcriptase has the job of converting the viral RNA into viral DNA. Now that the viral DNA is made, it must be inserted into the host cell DNA with a process called integration. As you can see in the leftmost figure, you have your viral DNA, which is attached to a protein called integrase, represented IN. The integrase chops off part of the ends of the viral DNA, creating two sticky ends so that the host DNA can be joined. In the third figure, you have your final product after the host DNA has been split in half and the viral DNA was inserted into the middle, creating one long strand of DNA with your incorporated viral DNA.
The central dogma explains that DNA codes for RNA, which in turn codes for functional proteins. HIV virus can go back and forth from RNA to DNA, then back to RNA, then to proteins. Once a virus particle invades a T cell, it releases its genetic material in the form of RNA, which is then made into double-stranded DNA in a process called reverse transcription, so it can be integrated into the host cell's DNA. The host cell DNA, now containing viral DNA, is then turned into viral RNA in a process called transcription. The viral RNA can then go and infect other cells.
The host cell is used to make many copies of the HIV virus using transcription. Some of the RNA is used to make proteins that are needed to infect a new cell. Other RNA is selected to be the viral RNA. Two copies will be taken to a new cell, turned into DNA, and inserted into the host genome. The RNA and proteins that are needed to infect a new cell must be packaged in order to leave. They are assembled into individual compartments called virions. In the diagram, you can see the proteins and viral RNA that have been produced. The proteins bind to the RNA and are brought to the plasma membrane. Other proteins, called envelope proteins, which are needed to construct the virion, also localize at the plasma membrane. Once the viral components have been assembled, it is ready to bud off from the host cell. The virus uses the host cell's plasma membrane to form its compartment or virion. Inside are all of the proteins and the two copies of viral RNA that are needed to infect a new cell. The outer shell of the virion has many docking proteins called GP120. These help the virus to find a new host cell and fuse with its plasma membrane. When the virus has been pinched off from the host cell, it is called an immature virus particle. If the virion stayed like this, it would not be able to infect a new cell, and the HIV virus would not be able to spread. Keeping the virus immature has been a large part of drug development. In order for the virus to become infectious, it must go through the maturation process. In the virion is a protein called protease that cuts up the non-functional polyproteins. Once the strands of linked proteins are cut into individual proteins, they can fold up into their functional forms. This is an important step because if the proteins did not work, the virus would not be infectious. When the protease cuts up the polyproteins, it also releases proteins that form a core capsid around the viral RNA and the proteins that are needed to get the viral DNA into the host genome, such as reverse transcriptase and integrase. The capsid is formed so that when the virion fuses to the new cell, the RNA can be easily delivered to the nucleus. Once both of these steps have happened, the virus is infectious and will fuse to a new cell to start the replication cycle over again.
There are 6 current classes of HIV drugs. The NRTIs and NNRTIs work by inhibiting reverse transcriptase, thus preventing viral replication. Without reverse transcriptase functioning as it normally does, the viral genome is no longer transcribed into DNA, and thus no proteins can be transcribed. The protease inhibitors act by inhibiting the function of protease, which eliminates the maturation process in the viral particles, so that is how those drugs work. The entry and fusion inhibitors prevent the entry of the virus into the cell either by blocking CD4 binding to the HIV particle or by preventing the membranes from fusing together. And then finally, before a doctor can prescribe any sort of treatment, they must perform a patient assessment on anyone with HIV. The patient assessment generally follows determining the viral load, the T cell count, and whether or not they have a resistant strain of HIV. The viral load is defined as the amount of viral particles in the bloodstream, and the T cell count is the number of living T cells in the body. A successful treatment works by decreasing the viral load and increasing the T cell count, as this is what is the normal situation. And one final consideration is that they must determine whether there's a resistant strain of the HIV so that they don't prescribe a drug that would not be effective. 3 other factors to consider when determining a treatment plan are the drug dose and administration, the interactions with other medications, and potential side effects. The reason why drug dose and administration needs to be considered is that if the patient is required to take many pills a day, they're less likely to comply and take their pills in the scheduled amount of time. Additionally, the interactions with other medications that they may currently be taking to combat opportunistic infections that go along with AIDS need to be considered due to possible harmful side effects. And then the drugs by themselves also have problematic side effects ranging from headache to nausea and then to more severe side effects such as kidney and liver failure.
We have seen that there are many effective treatments for HIV. However, unfortunately, HIV is a very hardy and adaptive virus, and if each one of these treatments is used individually, HIV will quickly gain resistance to it, and the drug will no longer be effective. Therefore, most HIV drugs are given in combination therapy with multiple types of drugs. Giving multiple drugs at once has both positive and negative effects. The positive effects include increased effectiveness because the drugs can work together, and reduce resistance because multiple drugs will target different elements of the virus. However, there are also more pills, more side effects, and a higher cost associated with the combination therapy. A new class of multi-class combination drugs that are one pill that contain multiple drugs have been given a lot of research money in the recent years, and these have the benefits of reducing pill burden and side effects from combination therapy.
Vaccines are important preventive measures for disease. A vaccine contains a weakened organism or a dead virus that is injected into the human body. This virus, or this dead virus, contains a surface antigen, which basically is how the body recognizes it as a foreign substance. A cell called a B lymphocyte interacts with this foreign substance and then evolves into 2 different types of cells. First is the plasma cell, which is responsible for creating antibodies, and these antibodies will bind with the foreign substance and target it for destruction. More importantly, however, is the creation of memory B cells. These memory B cells have specific antibody binding sites, which will target any antigen in the future that it has already encountered. So here is a list of vaccine trials that people have been attempting. You can see that there are multiple different ones, and this is because the vaccine is it has high mutation rate. This means it's kind of like when you have a cold and you have to get a new vaccine every year because the cold mutates and the vaccine from the previous year no longer works. In this case, this is the same case as HIV, and this is why you have to have so many different types of vaccine trials.
So now that we've talked about the science of HIV, how it's transmitted, and some of the drugs that are used to combat it, it's important to talk about who is at risk for contracting the virus and some ways to reduce the risk of contracting the virus. So just about everyone is at risk for being infected with the virus through sexual contact. That includes heterosexual couples, as mentioned before. Men who have sex with men are obviously at a very high risk for contracting the virus because of anal sexual contact. Women who have sex with women are also at risk because vaginal fluids can communicate the virus. And obviously intravenous drug users are at risk if they are sharing equipment. I'm now going to be going through some techniques for reducing your risk of HIV infection. It's important to note, though, that it is impossible to completely eliminate the risk of infection if you're going to be participating in any sexual contact whatsoever. There's no such thing as safe sex, there's only safer sex. The only way to ensure that you will not contract an STI like HIV through sexual contact is to not have sex in the first place. But this is generally not a practical solution for most people. The next best option is to always use condoms whenever any sort of penetrative sex is going to be happening. This includes oral sex, although HIV infections orally are very rare because the CDC website has mentioned other sorts of STIs like chlamydia and gonorrhea that can actually infect your throat if you swallow something like semen or vaginal fluids, which can communicate these sorts of STIs. So condoms or dental dams can be used to protect yourself orally. When having vaginal sex, pregnancy is a risk, but so are STIs like HIV and chlamydia and gonorrhea, and so female and male condoms are both available to protect you. Anal sex is a very high-risk activity because the anal canal is very easily damaged by penetrative activity in both men and women. So it is highly recommended to always use a condom whenever you're having any anal sexual activity. Finally, be very careful when using drugs and alcohol because these can lead to risky and unsafe sexual activities. It's important to note, however, that it is possible to have safe and low-risk sex with people who have HIV. As I mentioned before, it's always important to use condoms. However, there are medications available called pre-exposure prophylaxis that are basically HIV medications that people who do not have HIV can take to reduce the risk of infection. People who have HIV can also keep their own infection under control by lowering the total viral count in their blood by taking HIV inhibitors, like the drugs mentioned before. Finally, if you are involved with someone who has HIV, always make sure that you are continually being tested just so you know your status and if you contract the virus, can get treatment right away. If you are involved in using any sorts of drugs that are injected directly into your bloodstream, it's important to never share equipment with others because this is one of the highest risk means to communicate the virus. So that includes clean needles, cotton, and rinse water as far as equipment is concerned. There are clean needle programs available all across the country. However, some areas do not have them in as established a fashion as others, like Colorado Springs. If you do not have access to a clean needles program, you can sterilize them yourself by a thorough rinse with something like rubbing alcohol and then sterilizing it with a flame. There is also drug and alcohol helplines available to help you if you have a problem. The big takeaway here is to always use a condom if you're going to be participating in sexual activity and to know your status. There are many places where you can get free HIV testing available, and these are designed so that people can know their status and receive treatment for HIV infections as soon as possible.
We wanted to provide additional information. We mentioned the importance of testing, and the website provided gives information to Southern Colorado AIDS Project, their address, and phone number. They provide testing from 9 a.m. to 4 p.m. Monday through Friday, as well as special occasions. In terms of telling others, the website provided gives general guidelines as to when it's appropriate to tell others, and for example, if and when to tell employers or people you're dating, as well as family members. In terms of connecting with others, this website provides it's multiple conversations, sort of a thread that you can talk to others who are going through what you're going through. Whether you have a family member affected or just a friend, or you yourself are affected, you can find a conversation to connect with people. Nutritional supplements with HIV, the website provided gives additional information as to nutritions and supplements that are needed. Living with HIV, you often don't get as much nutrition as needed, so this website provides that information. The Symptoms of Early Stages website takes you through the stages of infection and what to expect at each stage, so you know if how you're feeling is normal or if you should confront your doctor. Side effects of medications is actually a chart that lists all the available medications and the side effects. So again, you can decide if you want to take certain medications and how you should be feeling when you are on those medications. In terms of financial support, AIDS drug assistance programs provide financial support because these drugs are so expensive. And we have listed a website that gives background information as to when this program was founded and who's received assistance in the past in terms of age, race, gender, and income. There's also a phone number provided to find the actual application online. In terms of medical news, the website provided gives the most recent stories or news, some science, and some more personal stories.
We hope that you now have a better understanding of HIV as a virus and how to prevent its spread. We would like to thank all our sponsors and Colorado College for making this talk possible.